Mass spectrometry is an analytical technique used to identify molecules by converting them into ions. These ions are then separated according to their mass-to-charge ratio, also called M/Z, and finally detected to produce a mass spectrum. First, the sample is introduced into the instrument under high vacuum. This vacuum is important because it prevents ions from colliding with air molecules. Next, high-energy electrons, usually around 70 electron volts, strike the sample molecules in the ion source. This process removes an electron from the molecule and forms a positively charged molecular ion. The general reaction is M plus electron gives molecular ion plus 2 electrons. After ionization, the molecular ion may break into smaller fragment ions because of the extra energy it receives. These fragments are very useful because they help identify the structure of the original molecule. Then, the charged ions are separated according to their mass-to-charge ratio. Lighter ions and heavier ions behave differently, so the instrument can separate them. Finally, only charged ions are detected and recorded. Neutral fragments are ignored because they do not respond to the electric or magnetic fields inside the instrument. So, the full process is ionization, then fragmentation, then separation by M/Z, then detection, and finally a mass spectrum is produced. The key point is that mass spectrometry does not directly see neutral molecules. It first converts them into ions, separates those ions, and uses their pattern to identify the sample. A molecular ion is the charged form of the original molecule. It is produced when the molecule loses one electron during ionization. This molecular ion is usually written as M plus DOT, which means it is a radical cation with an unpaired electron. The M/Z value of the molecular ion usually shows the molecular weight of the compound. So, it helps us know the actual mass of the original molecule. After formation, the molecular ion may contain excess energy. Because of this extra energy, it can break at weaker bonds. This breaking process is called fragmentation. During fragmentation, the molecular ion breaks into smaller charged pieces called fragment ions. These fragment ions are very important because they give information about functional groups, bond positions, and the molecular skeleton. The most intense peak in the spectrum is called the base peak. It usually represents the most stable fragment ion. For example, in alcohols, fragmentation may occur by loss of water. Ethanol can lose H2O and form a smaller fragment ion. In alkyl halides, fragmentation often happens at the carbon-halogen bond, such as the C-Cl bond. This produces smaller ions that help identify the presence of halogen groups. So, the key takeaway is the molecular ion gives molecular weight, while fragment ions give structural information. A mass spectrum is a graph that shows the relative abundance of ions against their mass-to-charge ratio, called M/Z. On the x-axis, we have the M/Z values of ions. On the y-axis, we have the relative abundance percentage. The base peak is the tallest peak in the spectrum. It is set at 100% and represents the most abundant and stable fragment ion. The smaller peaks are called fragment peaks. These peaks give clues about the structure of the molecule. The molecular ion peak, written as M plus dot, usually appears at the highest M/Z value and helps determine the molecular weight of the compound. Some compounds also show isotopic peaks. These peaks help identify elements that have multiple isotopes, such as chlorine, bromine, and sulfur. For example, chlorine shows an M and M plus 2 peak in about a 3:1 ratio, while bromine shows an M and M plus 2 peak in about a 1:1 ratio. So, the key takeaway is every compound produces a unique mass spectrum. By studying molecular ion peaks, fragment peaks, and isotope patterns, we can find the molecular weight, identify elements, and understand the structure of the compound,